The overall goal of ODELAY is to measure growth phenotypes of individual cells growing into colonies in a high-throughput manner.
This method can help answer key questions in microbiology, genetics, and systems biology, such as the effects of genetic perturbations and drug interactions on growth phenotypes of any colony-forming microorganism. The main advantage of ODELAY is that it enables the researcher to directly observe and quantify population heterogeneity amongst a large number of individuals.
Prior to assembling the agar mold, clean the acrylic molds with 70% ethanol and dry them using forced air. There are 3 bottom pieces and 2 upright pieces. To begin assembly of the agar mold, take the 3 bottom pieces and assemble them so that the 2 identical pieces sandwich the 3rd piece. Use 2 small binder clips to clamp the base pieces on each side. Place the uprights into the mold, making sure that the laser kerf is correctly positioned. Place a cleaned and dried glass slide on the mold and hold it in place while placing a second glass slide on the other side. Clamp the assembly with a larger binder clip. Clamp down both slides with larger binder clips, making sure each upper binder clip is in contact with the glass slide overlapping the acrylic. Before filling the assembled mold with molten agar, ensure the edges are sealed by pipetting about 70 microliters of molten agar media along the inner side of the mold. Fill the mold without trapping air bubbles inside by slowly pipetting the molten agar along the edge of the mold.